So let's pick up where we left off before. We're now going to begin to ask some questions on the previous material that I showed in part 3. And we'll start off with what types of cells do we find in connective tissues. And the textbook example, of course, is literally the textbook example we use inside your textbook is areolar connective tissue. You should look at that figure and go, how would I see these cells on a test question? And that's pretty simple. Imagine you're pointing at them, and I know inside your textbook there's a table that defines each of them for you, so you're going to go back and you're going to address that. But what I want you to pay attention to is more like question number 2. What is the physical difference between, say, dense regular connective tissue and dense irregular connective tissue? They're both made by fibroblasts. It's all collagen. Why are they in different angles? Where would you find them inside the body, and why is that important? Then if we take it one step further, maybe take a look at, say, adipose tissue itself, and then realize each of those, that population of cells beyond storing triglycerides has many more responsibilities than just storing connective tissue, meaning those triglycerides. What other responsibilities does it actually have? Okay, and that's what I want you to pay attention to. So we're going to move on now to the mature connective tissues, which we call collectively cartilages. And here you're going to see that cartilage is, of course, collagen. These are going to be chondroblasts. Remember, all blast-type cells make site-type cells will derive, or site-type cells will derive from blast-type cells. And in this case, we're going to trap the mature cells. This little arrow right here. The mature cells are going to be the chondrocytes, but they're trapped inside the lacuna, but they all came from chondroblasts. And in development, they were surrounded by this structure that's made of dense, irregular connective tissue that had blood vessels and nerves at one point in time, but no longer does. What I mean by that is part of the thing you want to pay attention to while you're reading about these different types of cartilages is what happens to them. I mean, why does Mother Nature put them where she puts them? What's so significant between, say, hyaline cartilage, elastic cartilage, and fibrocartilaginous cartilage? I'm not even quite sure that makes sense, but whatever. Okay. Hyaline cartilage is the easiest one. It's the most abundant inside the body, surrounded by perichondrium in development, and it's responsible for flexibility and support. I write here it's to reduce friction, but if you think about what actually happens at the ends of your bones, you jump up and down. This actually ensures that there is no direct friction between the ends of the bones themselves. You think about elastic cartilage. Perichondrium shouldn't be present. You know this because any damage to those tissues means that the cells themselves die and the tissue itself never really returns to its original shape. They're within a thread-like network of elastic fibers, but also inside those elastic fibers is some collagen. You'll see the lacuna, or inside of the lacuna are these cells that are chondrocytes. You notice there's a different sort of ratio. It looks like it's 1 per. It gives these tissues its strength and elasticity. Remember, inside class, I had you twist your ears so you could see it went back to its original shape. We're looking at fibrocartilage. You have chondrocytes that are scattered among bundles of collagen here. And remember, the cells made the collagen. They trap themselves inside these lacuna, and you'll see the wave-like structure has different numbers of cells. So you have different numbers inside the lacuna, and you see this wave-like structure, and that's collagen inside the matrix. And that matrix is going to give us the strongest type of cartilage that's available, and that's why you find them at, say, your intervertebral discs. One thing I want to point out to you is that, as I told you a moment ago, it's going to repair slowly. Okay? That's because it's avascular as it's developed. We've gotten rid of most of the actual tissues support by removing vasculature. There's some surrounding it a little bit, but on the whole, the diffusion of waste out of those lacuna to blood vessels is at a great distance. You actually have 2 patterns of growth. We don't really get into inside this class all that much, but you have interstitial growth, which is from within the tissue, and appositional growth, which will be from the outer surface of the tissue. So you can see how your cartilage can grow in size, and it's going to grow in 2 distinct patterns. We're going to move on now to bone tissue. Bone tissue, of course, is hard, which means the blast-type cells are going to give rise to an intracellular, or between extracellular matrix. Sorry, I said intra, but I meant extra. Extracellular matrix that stands between each of the cells. So osteoblasts make their own matrix. It is collagen and the actual salts. The salts are the calcium and phosphate salts, and what's going to happen, of course, is this makes bone, it's resistant strength. It's actually very much like a chalk, if you will. A dead bone is very much like a chalk, where you have that salt inside this sort of yellow space here. And you have the live cell, which is the osteocyte, which sends processes between each of these structures that are called canaliculi. But the cell body is inside, again, a lacuna. You have two types of bone. You have compact bone or spongy bone. And you can have a repeating system. And you saw this inside lab. This repeating system is, here's the Haversian canal. It'd be called the central canal or something else because they're getting rid of names inside your textbooks. But you can actually see that the pattern here is radiating outward from where you have blood vessels and you have nervous system structures and lymphatic structures. Okay. The second to last type of tissue we're going to talk about are muscular tissues. Remember when I drew the structure on the board, I had skeletal, cardiac, and smooth muscle tissue. They all use ATP to generate force, and they have, of course, obviously different types of functions. So if we think about how it is we're going to regulate how that cell functions, right? You actually have that skeletal, you have cardiac, and you have smooth muscle. And all of those are going to just simply, they cause the contraction of tissue, right? But to do that, they actually have to use ATP for that energy force. Okay? So you don't make all of this up. We're going to see as we go through the semester that this is what's going to actually be used to shorten the distance between 2 points. Remember, A pulls towards B and B pulls towards A. Let me see if I can draw that out for you here. Remember, inside class, what I did is I drew you these two spots here. All muscular tissues, skeletal, cardiac, or smooth muscle, all do the same thing. They pull towards the central structure here to shorten the distance between 2 points. Okay, so if you can understand this, you know it's going to utilize ATP to do all of this work, even though we have 3 different types, generally speaking. Hopefully you leave chapter 4 understanding that that's what muscle cells do. Skeletal muscle tissue, it has a repeating pattern here. You can see that we don't tell you how yet, but it has multiple nuclear structures, and they're all peripheral. And what you do is Mother Nature creates these lines of striations, and this is for linear muscle development. If you think about how muscles work, they're just a series of straight lines that are pulling on different places. When you're thinking about how or what Mother Nature is going to do here is she pushes the nuclei out of the way to make sure you actually have the straightest line. I'm sorry, that's not a straight line. Straightest line between 2 points. They're actually pulling on one another. Okay? Cardiac muscle tissue, when you put them together, they actually have sort of angular responsibilities. You have a single centrally located nuclei inside each cell. You still have contractile fibers, but the sum of these is they create angles, and because they create angles, it actually causes the cells to contract in a sort of squishing motion. It's a twisting, squishing motion. But each one of the cells is attached to an adjacent cell using intercalated discs, or intercalated discs. And remember, these, from the beginning of the chapter, are made up of the protein connexin. So the protein connexin actually makes up gap junctions, which means every single one of these cells has a gap junction alongside it, and that gap junction ensures that ions can pass in between the cells themselves. Okay. Smooth muscle tissue. We spend the least amount of time thinking about this inside AMP1, which is kind of a mistake because all of AMP2 is all the smooth muscle tissue that's regulated inside your body. We spend our time thinking about it inside chapter 15, and that's far, far away from tonight. But to finish up this chapter, I want you to see that they're part of your involuntary control, which is part of your autonomic nervous system. Find them in our blood vessels, our airways of our lungs, stomach, and intestines, which means we don't think about what they're doing when they're doing it, but they're being told what to do. A subset will have gap junctions, but on the whole, they slow muscular control and therefore slow relaxation of that control as well. Nervous system tissues consists of two principal types, and you'll see when we get into chapter 12, we break up the neuroglia to their responsibilities. These are the non-excitable cells that are collecting information, but on the whole, they are supporting. Neurons are the excitable cells, and you might recall inside class I drew pluses on the outside, minuses on the inside of the membrane here. Notice I said membrane, not the entire cell, and that allows the cell to become excitable. Remember, this is the dendrites in the cell body, so we're collecting information here. We're going to sum all of that at the axon hillock here, but then if we have that all or nothing principle activated, we're going to send that information, this is not even on this cartoon, to the axon terminals. They're excitable. Neurons and muscular cells are excitable, which means they have to generate some form of an action potential. Let's see if I can do this for you. Action potential. Okay, good, it did bold. Those action potentials are the general idea where we change that, temporarily change that charge at the surface of the cell. For muscles, it's going to be muscle contraction. For neurons, it's going to be released neurotransmitters or not released neurotransmitters. And this is actually how excitable tissues work. They have a specific responsibility, and our job is to figure it out. How do we actually repair all of these tissues? Can we repair all of these tissues? Well, in some repair, fibroblasts divide rapidly, but they need blood capillaries to keep them alive. So you can see for maybe areolar connective tissue, it repairs quickly, but say collagen, it does not, meaning the collagen made inside cartilage does not repair quickly. Bone requires a certain amount of time to repair itself. But if we're going to repair it, we have to create this structure that's called granulation tissue. And obviously, as we age, this is going to slow down. So if it's slowed down, what's going to happen next? Well, we want to think about as tissues heal, they're going to do it better when we're younger, obviously. That's because tissues, when we're younger, have better nutritional states, but they also have higher metabolic rates. Think about the rate at which we repair proteins inside of our cells, because ultimately that's what we're going to do. In the end here, as we age, our use of glucose is going to decrease. The amount of collagen we make is going to decrease, and therefore the amount of elastic fibers as well that can be produced will decrease. And in the end, just look back. I mean, one way to think about studying for this is take a look at any one of the cells we learned about inside chapter 4 and say, what would happen if I aged? What would it actually look like? Okay, and that's a good way to actually review. Okay, and what I mean by that is, what did a specific cell do? How would, say, a neuron work at 85 years of age versus 15 years of age? Okay, so this is the end of chapter 4. I'm going to move on to chapter 5. I won't ask any more review type questions here, but you get the general idea what we're going to be doing. So thank you for your attention. Bye.